Hey there guys. So right now what I want to do is introduce one-way analysis of variance to you. We're going to go at it from some familiar territory. So again, this is Intro to One-Way ANOVA for Bio 321 students. You've probably written your proposal at this point, and now what we want to do is kind of get you up to speed on the analysis you're ultimately going to do down the road with your data. Many of you are doing two-way factorial designs, and you'll ultimately do two-way ANOVA, but I'm going to save that for a separate video. I want to introduce, first of all, just analysis of variance. And as I said, we're going to approach this using some familiar territory. So let's review a case where we have two groups that we're comparing, and we have some continuous variable, and we've gotten a sample of each, and we've got means plus or minus standard errors plotted, right? We always plot some kind of a measure of the standard error. Typically what people do is they plot plus or minus one standard error, and that allows you to look at the overlap of the error bars, but you can't really rely on that to do a statistical test. So as you recall from the last video, what test do? We do a t-test on something like this. And let's kind of review what that t-test would tell us, okay? So every statistical test should be answering a question. So really what we want to know is, what was the question that we were answering by doing the t-test? Well, the question had to do with the means of these two groups, right? Does mean y differ for groups A and B, whatever they happen to be? And remember, these groups are levels of a factor. So we have some kind of factor, and it has two levels, A and B, and those two levels produce means, and those means we are asking, are they different? Put a question mark. Does mean y differ for groups A and B? But I want to actually rephrase this a little bit. Remember what we got out of our analysis with two groups? We got a t-value. For example, we might have gotten a t-value of 12.831, okay? And I'm not going to go through the t formula here, but if you don't remember what it was like and how we got it, you might want to look it up. As I referred to last time, you can look at the Khan Academy or something. And we got a p-value associated with that t-value, okay? And so corresponding to the t-statistic is a p-value, which helped us interpret this statistic. Okay, so we would get something like a p-value of 0.013. All right, depending on our sample size and everything, we will get various p-values. So when we get this t-value, what question is that answering? All right, so we can rephrase this question a little bit. What is the chance we would get a t-value that large or larger when in fact there was no difference between mean y for levels A versus B. Okay? So, in other words, this no difference criterion that's sort of the null hypothesis. There's no difference. And what we want to know is, what is the chance we will get a t-value this large when in fact there was no difference? That is, the null hypothesis was actually true. And this p-value quantifies that chance, okay? So, in general, we say if p is less than 0.05, we say y bar A differs from y bar B. That is, the two means do in fact differ. In other words, we reject the null hypothesis that y bar A equals y bar B. Now you might say, why do we reject it when p is less than 0.05? We reject it by convention when there's less than a 1 out of 20 chance that we are actually making an error. And by the way, this is actually quantifying the chance that we are making that error. That's called the type 1 error rate. What do we mean by type 1? That means we're saying there's a difference, when in fact y bar A equals y bar B. In other words, the null hypothesis is true, and P is quantifying the chance that we are incorrect in saying there's a difference. So we are incorrect 1.3% of the time. 1.3% chance is our type 1 error rate, as indicated by this probability of 0.013. All of this has to do with how we interpret our t-test. So let's go back. What was the question? Does mean y differ from four groups A and B? We're comparing two groups. We do a t-test. We get a t-value. We get a p associated with that t-value. And that tells us whether we can reject the null hypothesis of equality of the means for the two groups, or whether we cannot safely do it. And our cutoff for safely doing it is when p is less than 0.05. And we then say there is a statistically significant difference between y bar A and y bar B. All right. Now, why 0.05? I said it was the 1 in 20 chance. You know, if it's less than 1 in 20, we're okay with saying there's a difference. We're kind of sticking our necks out, but not very far, because 19 out of 20 times we'll be correct in making that inference that y bar A does not equal y bar B. All right, so that criterion is actually called our alpha level. It is our cutoff level, and it's just a convention that we set alpha equal to 0.05, and we test p against that, right? We say p less than alpha means statistical significance of our statistic, whatever it is. Okay? In this case, it was t, and we saw the larger t, the lower p would be. Okay. All of this you've had before, but I wanted to kind of review what a p-value means. It is what allows us to detect statistical significant differences. Okay, let's move on to ANOVA. So what's the difference between ANOVA and t-test? Let's focus on ANOVA, and we're going to now talk about one-way ANOVA here. So I'm having, just to clarify the difference between t-test and ANOVA, I'm going to draw a diagram where we have three levels of something, or three groups that we're comparing, and we're looking at y bar. You know, we're going to put standard error bars on it again. So we're kind of jumping ahead to our results, and we're kind of thinking, okay, we're going to plot our means like this, and let's think about what question we are asking here. Now, we aren't asking whether A differs from B, or whether B differs from C, or whether A differs from C. We are kind of asking, are any of the groups different from any of the others? But there's another way we can restate it, just like our second question before. What is the chance that two or more group means, in this case 3, could have been obtained when in fact they were all drawn from one population. Okay, now in this case, our statistic measuring the variation among means is not t, it is f. But like t, the bigger it is, the more significant it is. That is, the lower p is. So for example, in this case, we can see some pretty big differences here. We might get an f value of 7.131 from our statistics program. And by the way, we're going to let SAS JMP calculate these things. I'm not going to make you do this by hand. If you take biometry class from me later, you'll get to do that. But for this class, since it's not a statistics class, we are just using statistics to answer questions, we'll let JMP tell us a p-value associated with this, and we'll see something like this. You know, we'll get a p-value, for example, of 0.0313, and we will have to interpret the significance. So I'm asking you right now, is that p-value statistically significant or not? What do you think? Yes, it is, right? Because we would generally let alpha equal 0.05, and we're going to compare p to that. Is p less than alpha? Yes, it is. P is less than 0.0313 is less than 0.05. And so we say, yes, this is statistically significant variation among means. There is significant variation among group means. Okay. And that is what ANOVA is telling us, whether there is or whether there isn't. In both of our cases so far with the t-test and ANOVA, we had statistically significant differences. Okay. Great. So now we know what an ANOVA is. It's asking, is there any variation among means? Is that enough? Well, we kind of would like to know which means differ from which other means, right? So we would then do something called an a posteriori test. So an a posteriori test sounds like somebody's rear end, right? A posteriori, your posterior. Okay, what it means is after the fact. So after you get a significant variation among means, you might wonder which means differ from which other means. And I'm going to show you in SAS JMP a simple a posteriori test called Tukey HSD, honestly significant difference test, which will tell me which means differ from which other means. And typically we would see something like this. We would see that we would put letters over those means that were different, and they would be different letters if they were significantly different. So in this example, group A is not different from group B or group C. It has the same letter as both, but group B differs from group C. Okay, so this is how we designate which ones are different. But I'm going to show you in SAS JMP how we get that information out. All right, so I think it's time to move to an example of a problem. So let's say we have set up an experiment where we have 5 replicate flasks in each of 3 conditions. Okay, and in these flasks we have put one of 3 treatments. 0 MCHM, this is our wonderful chemical that spilled into the Elk River downstate recently. We have another set of flasks where we have 0.5 grams per liter of MCHM, and then we have a high level of 50 grams per liter of MCHM. Okay, so we're going to call this a 0 level and a low level and then a high level. So these are our 3 levels within our factor, which is chemical pollution. All right. Now, we obviously need to be measuring some organisms in these replicate flasks, so what we're going to do is we're going to put in a Daphnia in each flask. And on the Daphnia, we're going to be able to extract them, and we're going to actually measure heart rate. By the way, this is something you can actually do as an interesting dependent variable. The Daphnia are sort of translucent. You can see their heart beating in there, and you can count it. It's a little tricky to do because they can have beats of up to 300 per minute. But this is possible. So we're going to have 5 replicate flasks. We're going to just extract 1 Daphnia from each flask, and we have 3 levels of this treatment for MCHM, or dastardly chemical, put into the water by Freedom Industries. Okay, now what we want to do is jump to SAS JMP. All right, so I'll pull up JMP here. And we double click on the little jumping guy down here. And by the way, you're going to get practice at this in lab, don't worry. And when you hit new data, up pops the data sheet, and I'm going to spread it out here. Okay, and this is our SAS JMP spreadsheet. Now, we think about our experiment. We have 5 flasks, right? We have 3 treatments. So we're going to need 15 observations, 15 rows of data. There are 15 Daphnia. So you can go up to rows, and you can add rows. And it will let you add, thought it would. Let me add rows. Ah, there we go. Add rows of data. I'm going to type in 15 and look at that. All right, so now in these columns of data, so we have sort of our 15 observations we're going to make here. We're going to need to put our independent variables and our dependent variables. So what is our independent variable in this case? It's chemical concentration, right? Or chemical treatment. We'll call it treatment for the purposes of this. Okay? And I have a 0 treatment. We have 5 flasks of the 0 treatment. Oops. I don't want to do that. I want to have caps lock on. And I have 5 flasks of the low treatment, and I have 5 flasks of the high treatment. Okay. Now, so that's my independent variable. I also need my dependent variable. So I'm going to click on the column here, and it creates a nice new column. I'm going to double click on that and type in heart rate. All right. Now, heart rate is going to be a numeric variable. It's continuous. Okay. And I am just going to make up some numbers here. I could have picked an easier thing, couldn't I? So let's make up some numbers. Let's say that we have, oops, heart rate of 1 is not too likely. And I'm kind of, imagining some variability here. Oops. I guess I'll need to get my glasses on for you and look really nerdy. Okay, now we have our low treatment. And then we have our high treatment. All right, so our question is, has treatment affected heart rate? And in fact, generally, when we're doing these, what we're doing is we're creating a model. This is called a statistical model, the ANOVA, where we have our dependent variable as a function of our independent variable, and we're asking, is there a relationship between the two? does level of treatment here, the Z versus L versus H, affect heart rate? All right, so let's go to our analysis menu before we run out of juice in my machine. And what we're going to do in this platform, we're going to use fit Y by X. Notice it says, pops up, how a response distributes differently across a column factor. That's kind of obscure, but it's general. And we want to actually look for these little bars. That indicates that this is a class variable. We have 3 classes of treatments, and that's going to be our X. So we're going to cast it into the role of X variable or independent variable. And we have heart rate, which is continuous, indicated by this triangle, and we're going to put that into the role of a Y variable. So what we're asking is, does the response, is it affected by the factor treatment? Okay, we hit OK, and up pops just a plot of some results here. Not even very meaningful. It's not even a nice histogram, right? So we haven't really done any analysis yet, but if we go up to this little tab up here, we see lots of options. And what we want to do is do a means ANOVA. So this is actually plotting means and some measure of the variability here. And as we go down, open this window, we see an analysis of variance. Okay, so this, in fact, is the heart of our analysis of variance. Now, C total is not normally put into an ANOVA table, but if you were asked for a complete ANOVA table, you would give all of this: the source of variation, degrees of freedom, sums of squares, mean square, F ratio, and p-value. And that p-value is associated with that F value. Okay, so we see, and we would also put the error term in, the error row. In fact, if you look closely, you can see what the F ratio is. The F ratio is the mean square, that's another name for the variance, by the way, associated with treatment. Hi, bird. It's a robin. It's not robin-like weather out there, is it? But the mean square for treatment divided by the mean square for the error is actually the F ratio. So this F statistic is a ratio of mean squares. One of them that contains the treatment differences and one of them that doesn't. Ooh, we're starting to get into the nitty-gritty of what F tests actually are. So the p-value, though, that's what we want to look at to judge whether we could have obtained an F value that large or larger by chance, when in fact there's no difference between means, and our chance is incredibly small. So we have a very small chance that the heart ratio, or the heart rate of these Daphnia is not being affected by the treatment. It definitely is being affected by the treatment. We can state that with a high degree of certainty because p is less than 0.0001. Now, which means are different from which? I mean, you can look at it and see, right? But we can actually compare means. And I told you I was going to recommend the Tukey HSD. It's also called the Tukey-Kramer HSD. But anyway, we'll go Tukey HSD, let that up, and look for the letters down here. Okay? This is called the Connecting Letters Report, and this tells which group differs from which other group. So this is a little different from the example I gave you before. In this case, every mean here, every group, is significantly different from every other group. Okay, so that's one possibility as an ANOVA result. But we would, again, draw a nice histogram of those results and show the letters on top of the histogram. There we go. We have our one-way ANOVA. I'm going to do a little separate example for the genomics people on linear regression, but FYI, it just uses the same platform. Okay, so there's your one-way ANOVA example. Be ready to practice that in class, and then we're actually going to have a quiz on it in class as well. Oakley dokley. See you later.